All right, so looking at cell structure and function, key concepts, we know that cell is the basic unit of life. So again, focus on not basic molecule or basic unit of matter, it's the basic unit of life. So all living things, the smallest component of that living thing is the cell. And it determines the function of the body as well. Plasma membrane, cytoplasm, and organelles are what all cells have in common. We talked about that in lab last week as well. One thing I do want to mention is the cytoplasm is not just the fluid, but there's also like a scaffolding made up of tiny little proteins that give the cell its shape and structure, so it holds the cell. You know, the organelles can embed themselves in that cytoskeleton. And cytoplasmic inclusions are little chemicals that are floating around the cytoplasm. For example, melanin is a pigment in our skin cells that give your skin, and it gives your hair and your iris of your eyes its characteristic color because of varying amounts of melanin. So melanin is not an organelle, it's called an inclusion. It's just a chemical that we find in cells that are not organelles. So definitely know the definition of what an inclusion is versus an organelle. So inclusions are chemicals, not organelles, and they're found in the cytoplasm. Organelles are membrane-bound structures found in the cytoplasm. We're going to talk about their functions today. So more examples below of cytoplasmic inclusions. Again, they're chemicals, all different types of things. Okay, the internal environment we already mentioned, we talked a little bit about before, and you have a worksheet that covers that in your packet. Functions of the cell, we'll go through those as we go through each body system. Remember, the internal environment is the fluid that bathes our cells. So the plasma and the interstitial fluid, the plasma is the fluid portion of blood, interstitial fluid is that fluid between cells. So it's where all the interaction is happening in the body. That's why it's called the internal environment. It's where cells are interacting with each other. So interstitial fluid and plasma make up extracellular fluid. And again, where is this found? Inside or outside cells? Outside cells. So definitely know these definitions and where they're located. Are these fluid compartments in cells or outside cells? There's only one fluid compartment inside cells, and that's the intracellular fluid, ICF, we said, right? So inside cells is where we find most of our body fluid. So this is a key concept here. Make sure you highlight, underline that, and know that for our test. So the plasma membrane then, it's selectively permeable, which means not anything can enter or leave the cell. It has to have certain characteristics to freely move, and we'll talk about that. So in lab, we talked about the different structures, and we'll talk a little bit more about that again. So when we look at the cell membrane, we call it a fluid mosaic model, which means that it's somewhat fluid because it has an oily foundation or base, and that's the phospholipids. Those are oily molecules. It's a type of lipid, and the heads the heads attract water. We say they're hydrophilic, which means that things are attracted to the surface. And then hydrophobic means the core of our plasma membrane repels water-soluble substances. So like we know water and oil don't mix, right? So if we have an oily core to our cell membrane, then anything that dissolves in water, like ions, sugars, are not going to be able to pass through that fatty core, those tails on those phospholipid molecules. So we have to have transporters for some of those things. And then cholesterol stiffens the membrane, so that, remember, that was embedded in our membrane, those little yellow structures embedded in the fatty core. That stiffens the membrane. So when we talk about the plasma membrane being made up of two layers, that's those phospholipid molecules. And again, the tails repel water and the heads attract. So the tails are hydrophobic, hydro meaning water, phobic meaning fearing, water fearing, and the heads are hydrophilic, meaning water loving. So they attract things to the surface because we want to get things to the cell surface, and then we have to decide how to get them across. We're going to talk more about that today. So some require special carriers if they are repelled by that fatty core. So the more cholesterol found in the membrane, the stiffer the membrane will be, and that's okay, but we know that we don't need too much cholesterol in our diet because it stiffens our blood vessels and makes them less elastic and can actually narrow the inside of our blood vessels, and that's a big reason for heart attack, stroke, and high blood pressure, is too much cholesterol in the diet from animal products like dairy, high-fat dairy like cheeses and meats. So we have to be careful with our cholesterol intake. So let's talk about some of these proteins. We have proteins embedded in the membrane. So again, here's our phospholipid molecule. There's the heads that are hydrophilic, and these tails, the core, is hydrophobic. So we have proteins that are embedded in the membrane, and some of them span the entire membrane, so we call those integral or intrinsic proteins, and others are just on one side or the other. Usually those are enzymes when they're just on one side or the other and they don't span the whole distance. So often these integral proteins act as transporters. Sometimes they have receptors for changing the cell's function, and we'll look at some of those examples. Some of these proteins are attached to a carbohydrate, and their job is for cell communication and recognition. So carbohydrates are really important for, that's what these structures here are, for cells to communicate and recognize each other. So if the carbohydrate is attached to a protein, we call it a glycoprotein. Other cases, we have these carbohydrates that are attached to the surface of phospholipid molecules, to the phosphate, the head, and then we call those glycolipids if they're attached to a phosphate group. So in this case, this is a glycoprotein because it's attached to a protein. The carbohydrate is. So the carbohydrate is this part that extends off the outside of the cell, and it's used for communication. So some proteins are channels. So we can see this one is open, and it allows water-soluble substances that can't dissolve in the fatty core. They can pass through here and get into the inside of the cell or outside the cell. So there's 3 types of channels, and you need to know the 3 types for our test. Non-gated means they're always open. Things can freely go from high to low concentration. Ligand-gated. Ligand is any chemical, just a fancy name for a chemical. So it means something has to bind to that channel before it's going to open up and let that substance through. So that's a little more selective, right? We can't just let anything through. If it's ligand-gated, it needs a chemical to bind to it. And oftentimes that chemical is acetylcholine. But there's other things it can be too. And then voltage-gated means there needs to be an electrical charge difference on the membrane in order for that channel to open. And we're going to talk about that a lot this semester. So there's non-gated, ligand-gated, and voltage-gated channels that allow things through the membrane. These are obviously selective, the ligand-gated and the voltage-gated, in order to let things across. And we'll talk more about those. And then here's an example of receptor molecules can be found on the surface of a protein. And in this case, we can see that acetylcholine has to bind to this receptor on this channel. What kind of channel would this be if it requires a chemical to bind to it? Ligand-gated. So this is an example of a ligand-gated channel. When acetylcholine binds, it opens up, and then sodium can pass through, because sodium cannot diffuse in this fatty membrane because it's water soluble. It has to go through a channel to get inside. So as long as acetylcholine opens the door, sodium can go in. So we need acetylcholine before sodium can move, and we'll talk a lot about that again, like I said, this semester. Some proteins are not channels, but they act as enzymes. So this is an enzyme that acts on the surface of the cell. Other enzymes are on the inside of the cell. But in this example, we have others that act as carrier proteins. They're not really channels, but they attach to a specific substance, and they change their shape and dump it on the other side of the membrane. So it's not a channel for flowing of substances, but actual grabbing it and moving it into the cell. So that's a carrier protein. But either way, no matter what the substance is, if it's a large globular structure that spans the membrane or is on one side or the other, we know that it's a protein, right? And there's different types of proteins. So proteins really determine the function of the plasma membrane. So if we go back to that first slide talking about proteins, So lipids, the phospholipids here, the cholesterol and the phospholipids, that determines the structure of the membrane. So make sure you highlight the structure. So the brick and mortar of the plasma membrane is the structure. It's those lipids. That's on page 56. So it's the structure of the membrane. And then proteins embedded in the membrane determine the function of the membrane. Whether it's a carrier, a channel, an enzyme, all the action of that cell is really determined by the proteins embedded in the membrane. And remember, DNA is what makes those proteins, so that's why DNA is so important. It determines the function of our body. So proteins are the function of the membrane. Lipids make up the brick and mortar or the structure of the membrane. And we talked about carbohydrates are for marker, for cell identification. We talked about receptors, enzymes. So let's move into movement through the plasma membrane. There's a couple of different types of movement through the membrane. So this is on page 62. You've all heard of diffusion before, right? If I sprayed some stinky perfume up front here, it wouldn't take long before it'd be in the back of the room, right? Is that bottle of perfume alive or living? Is there energy applied to that perfume? No. It's just random movement of molecules. It's the law of conservation of energy. All matter is in motion unless acted upon by an outside force. So it's matter just constantly moving, just random movement of particles of a gas, and they eventually spread out until they fill the whole room with the mild smell of that stinky perfume, right? But it's strongest at the source, and then it diffuses away. So diffusion does not require energy. It's just a natural process, and we'll go into this in more detail on other slides, so you don't need to write it here. Osmosis is just like diffusion, but osmosis is diffusion of water. So we're just talking about water moving from where there's more water to where there's less water. And then filtration is due to pressure changes. So our kidneys are constantly filtering our blood because of our blood pressure. The higher the pressure, the more our kidneys filter. The less pressure, the less the kidneys filter. So what does that tell you about really low blood pressures? That can be not good for filtering the blood, and toxins can build up in the blood if a person has really low blood pressure. And if they're really high blood pressure, that can damage the kidneys because of those capillaries are getting such high pressure blood that they start to break down. So chronic hypertension is really bad for kidneys. Okay, and then we have these mediated transport mechanisms, which means there's some type of protein helper to get things across the membrane. So some things can diffuse right across. So you see in this diagram here, you see the little particles there, and they're diffusing right through with no help, just from high to low concentration. Those are things that are lipid soluble, which means they can dissolve in fat. Things that can dissolve in fat are alcohol, oxygen, thank goodness for that, right? We need oxygen to get quickly into our cells to feed our cells. Carbon dioxide, and some hormones, fatty hormones, those steroid hormones, like estrogen and testosterone. Other ones are like aldosterone, progesterone. Vitamin D is a fat-soluble vitamin, E, K. Those can diffuse right through with no help because they dissolve in oily substances. So that's why when a person comes to work and they've been drinking, you can smell it on them, right? Because it diffuses right off of their mucous membranes and through the, from the blood across their lung tissue and in their exhale. And that's why we can use a breathalyzer to determine how much alcohol is in the blood, because it's rapidly diffusing into the lungs and being exhaled. So we know that alcohol can diffuse. And another interesting thing is, not everybody knows this, but nurses do, and maybe you do. If you ever notice the breath that people have after a night of heavy drinking, there's a very specific breath that people have in the morning. That's a special aldehyde. As the body is breaking down alcohol, you know, a high amounts of alcohol in those early stages, they're blowing off not alcohol, but this unique smelling aldehyde. And I have been, you know, I've worked early shifts, the 6:30 a.m. shift on a Saturday or Sunday. And I remember one time particularly, I walked past an employee where I was working, and whoa, they were giving off that aldehyde big time. So they must have been, you know, out until 2, 3 in the morning and then got up at 6:30 to work. So Be aware of that. So if you do ever go out for a night of, drinking and you have an interview the next morning or you're going to work, make sure you're chewing some gum to control that odor because people recognize it. The informed people recognize it. Not everybody knows what that smell is, but some of us do. All right, so water-soluble things, they need a transporter. So they have to use some of these other types of transport, this mediated transport. They need a protein helper, and that's what these all use. Okay, so glucose, sodium, chloride, calcium, all those charged ions need help across the membrane. They cannot dissolve in the middle of that membrane. Another one I should mention here that a lot of people rely on is nicotine, right? People smoke nicotine. It diffuses through their membrane into their blood readily. So that is another lipid-soluble substance. And it's a big problem with medications. If we're trying to treat depression and anxiety, we need to get that pill down the digestive tract into the blood, diffusing into the brain to help people feel better and raise those serotonin levels, right? Sometimes we struggle with finding new and better medications because it has to be something that can dissolve across the membrane or have a transporter. Like we know marijuana has a receptor. The cannabinoid receptors that we have in the brain transports THC into the brain. Okay, so diffusion. Diffusion does not require ATP, so that's a key concept here. It does not require energy. It's based on random movement of particles. I spray my perfume, you smell it in the back eventually because of random movement from higher concentration to lower concentration. So it depends on how concentrated it is, right? If I did 10 pumps of my perfume, you'd smell more of it in the back. If I did 1 pump, you'd smell less, right? It'd be less concentrated with 1 pump on the spray versus 10. And viscosity is how easily, if we're looking at diffusion of a liquid, is how easily that flows. So for example, a highly viscous fluid, viscosity is a measurement of resistance to flow. So low viscosity means that it's very I better look. You know what? I'm struggling. I don't think I said that right. Viscosity. I thought it meant resistance to flow, but now I have to look it up. State of being thick. Yeah. Cooling. Yeah. Resistance to flow. I doubted myself there. But anyway, so highly viscous means that it's thick. So if a person doesn't drink a lot of fluid, and their blood is thicker, or they're really dehydrated, that causes less diffusion, metabolism is not as efficient, and we need to bring their fluid levels up because viscosity, high viscosity of blood and body fluids is not a good thing. So the thicker it is, the harder it is to flow. The thinner, low viscosity means it flows more easily. All right, so osmosis, like I said, is diffusion of water through a membrane. So there's 3 different types of solutions that osmosis can create depending on the direction. So it does not require ATP, and water always flows from high water concentration to low water concentration. So if a solution is hypotonic, it means it's very dilute. It's high in water. So if I'm making Kool-Aid and I taste it and it's really watery tasting, that's a hypotonic solution compared to one where I add a lot of extra Kool-Aid to and it's very sugary and sweet. That's a hypertonic solution. So it's low water content. There's a lot of stuff in there, a lot of solute, the stuff we dissolve in the water. So if a person is dehydrated, how would we describe their blood? Hypertonic, yeah, hypertonic. So if we put the IV pump to correct that dehydration, and then I walk away and I have it at like 100, or let's say it's flowing to gravity, it's going in as fast as the vessels will allow it, and then we forget about that patient and we come back at the end of a 12-hour shift, they've got too much fluid in the blood, then we would describe their blood as hypotonic, really dilute. That can be dangerous too. What about hopefully everybody in here today? What should your blood be? Isotonic. So you should have about the same amount of water outside your cells as inside your cells. We shouldn't see a big movement across the membrane of water if you're in a happy, stable environment, right? So things always move from high to low concentration until both sides are, what do you think? What? Equal, yeah. So that's the goal of these different concentrations. Water is going to move, and the solute is going to move until things are equal. So if I have an isotonic solution, my cells are neither going to shrink nor swell. They're going to be just content. Hypertonic, water is going to flow from, if I have a hypertonic solution, that means I have a lot of solution and not a lot of, I'm sorry, I'm saying that wrong. If I put a cell in a hypertonic solution, which way is water going to flow? It's going to go out of the cell because it's going to go from higher water in the cell to this concentrated solution that I just threw it in. So for example, if we take meat and we sprinkle salt on it, what happens to the meat? If you're making jerky, for example, water flows out of the cells of the meat, right, and forms a pool of fluid on the surface from the salt. So that's crenation. The cells will shrink. So we don't salt steaks before you put them on the grill. What they recommend is you put olive oil around the outside of your steak or your roast, then season it, because what does the olive oil do? Yeah, it creates a barrier. That water can't diffuse out to that salty seasoning you put on the outside, and it keeps keeps your cells moist. Because if we salt meat, it's going to pull all the fluid out, and then you're going to have a tough steak or a tough roast beef. Or what other people do is they sear it in a pan. When they sear the outside of it, that seals the outside of that steak, and then you can season it, and it's not going to lose it all. So, like when you throw something in a crock pot, if you throw it in a crock pot and put a bunch of seasoning on it, you're going to have a dry piece of meat unless you soak it in water and let it sit for like 10 to 12 hours, then it gets soft again. But anytime you put a cell in a concentrated hypertonic solution, it's going to lose water. So if I put a cell in pure water, water is going to flow from the hypotonic solution into the cell, and that cell is going to burst. But what happens is it doesn't burst because it moves back and forth until things are equal or isotonic, right? So, but cells can swell. If we keep pushing fluid and we keep having pressure outside the cell, it can swell a little bit where it's not ideal, and then people start to have symptoms if they have too much fluid on board. And that's a danger in kids that go to swimming class, mom and tot swimming classes. You let that child swallow a bunch of water, you know, they go under a little bit, or little kids don't know and they just consciously drink the water right when they're 1 and 2 and 3 years old. And pretty soon they're a little weird acting later on in the afternoon, and that's because their brain cells have actually swelled from bringing in so much fluid. So swim instructors really need to tell young children, like the 1, 2, 3, even 4-year-olds, they get a little smarter at 4. They know that, you know, my belly is getting full and I shouldn't be drinking this dirty pool water. But 3 and under, they don't really know, and moms need to be aware. Don't let your kids drink so much water. They can end up hypotonic in their fluid and causing brain swelling. Okay. So when we give fluids in the hospital, we want those fluids to be isotonic. So it goes in and we get just enough to equalize our body fluids. And that's usually the IV fluids are 0.9% sodium chloride is what we hang for patients that just need a little extra fluid because they're dehydrated. Or maybe you've gone in with a bad stomach flu or a migraine with vomiting. They're going to hang that sodium chloride. So here's just an example of the 3 types of solutions. Hypotonic, it's losing water because it's in this really concentrated solution. Isotonic, there's movement of water back and forth, but there's no net movement of water. Oops, I did that the wrong way. Sorry, this is the hypo. This is a very dilute solution, gaining water in the cell. This is a concentrated, maybe this is like a salt water, and water is leaving the cell, causing it to shrink. Again, what do we call this movement of water in and out of the cell? Osmosis. Water is not a living thing, so it does not require energy. Filtration, like I said, depends on pressure. And what type of pressure are we always talking about? We're talking about the body. Blood pressure. Yeah, blood pressure. That's the most common example when you look at how the kidneys form urine. So those osmosis diffusion through the membrane does not require energy. Facilitated diffusion means we need a transporter. We need a protein to help that water-soluble substance across the membrane. So facilitated means there's a helper, but we still don't need ATP. So again, you might want to highlight this. No ATP is required in facilitated diffusion. We're just going to take that substance. It's water soluble, so it needs a protein to get to the other side, but notice the concentration. High concentration to low concentration. It just flows down the concentration gradient, does not need any help. And this is a common way that glucose enters our cells, is by facilitated diffusion, because glucose can't get in, right? Because it can't dissolve in that fatty core. It's water soluble, so it needs a protein transporter. So again, highlight that. Glucose, that's the most common way that our sugar enters our cells, is facilitated diffusion. Does not require energy, but it does require this purple thing, which is a protein. Active transport, this does require ATP because we're taking something and moving it from lower concentration to higher concentration. Think of it like sliding down a slide. If I go down the slide from high to low, do I need energy? No. If I want to go from low to high, I want to go up the slide, that's going to take energy, right? I'm going to have to use my physical, mechanical energy to do that. So the same thing happens here in removing substances. The sodium-potassium pump is a ATP requiring pump that will actively pump sodium from inside the cell to outside the cell. And it takes potassium from outside to the inside. Notice the direction of the arrows. So the sodium-potassium pump is kind of like bailing water out of a leaky canoe. It's constantly taking the sodium, bailing it out, and then there's leak channels for sodium. And because there's more sodium building up outside the cell, sodium wants to leak right back into the cell. So the pump is constantly dumping sodium out, constantly bringing potassium in against their concentration gradient. So there's always more potassium inside the cell, and there's more sodium outside the cell because of these pumps. And these pumps are constantly active on all of our cell membranes, and we're going to talk a lot about this semester. So this is an active transport pump. Active meaning it's requiring ATP. And if ATP is not being produced because of a heart attack or some stoppage of blood flow that is needed to make ATP, our pumps stop. And if our pumps stop, our cells don't function and death occurs. And it all comes back to oxygen and ATP. There's a cardiac patient often is on medicines or have kidney damage, which often causes electrolytes to go off. And it can cause serious heart rhythm problems if electrolytes are off. Yeah, definitely. Secondary active transport means that you have, say, the sodium-potassium pump, but then you have another protein carrier that is going to use the concentration gradient created by another pump to drive the movement of another substance. So this is a little more technical. We're not going to go into great detail, but just know that two proteins are involved, and we're creating a concentration gradient using ATP from a different protein channel, but then this protein channel that is transporting glucose is not using ATP. It's kind of taking advantage of this one. So I kind of think of it like a taxi. So sodium is paying the taxi fare, and glucose is riding along because it's going from, sodium is going from high to low concentration here, and glucose is going from low to high, and it's riding along. And so this one does not require ATP, but this one does. And sodium wouldn't be riding in if it wasn't in higher concentration because of this pump. So it's not directly using ATP, but it's using another protein that is requiring ATP. So glucose, like I say here, is like a hitchhiker. It's riding along with sodium, but it's not directly using ATP. So we would say glucose can enter the cells using secondary active transport. So that's another way glucose can enter cells. The most common one, though, is facilitated diffusion. So other types of transport that are a little more basic, endocytosis. Endo means inside. So what happens is the plasma membrane engulfs solid particles and pulls it into the cell. So endocytosis brings it into the cell. These all require ATP. So there's different types. Phagocytosis means cell eating. So this is solid substances like bacteria. So a white blood cell that's attacking bacteria as part of the immune system, that's an example of endocytosis, specifically phagocytosis. When we see a lysosome breaking down dead organelles inside the cell, and let's say it's breaking down medicine that you had in your pills, your prescriptions, that process of breaking down, like the smoothie R, can wrap around those molecules and break them down, but they're liquid molecules. That is called pinocytosis, which is cell drinking. So phago means solid, so think of like chewing, and pino is drinking. All of them are examples of endocytosis, and they all require ATP. Receptor mediated just means there has to be a receptor before it takes it in. So here it binds to the receptor, then it takes it into the cell in the form of a vesicle. Exocytosis is when we're moving things out of the cell. So when something is produced by the rough ER, ribosomes, produced by the rough ER and then is shipped out of the cell, that's exocytosis, so it's exiting, leaving the cell. So digestive enzymes that are produced by pancreatic cells and then they enter the small intestine to do their work. Mucus produced by salivary glands enters the oral cavity. It's exocytosis, so we can bathe your mouth and esophagus, and then making milk by those cells inside the breast. It enters the milk ducts by exocytosis. So those are the means of transport. Which ones require energy? We talked about primary active transport, secondary active transport, and the last two things we mentioned, endocytosis, exocytosis. Yeah, those are the ones that require energy. Osmosis, simple diffusion, facilitated diffusion do not require energy. And they always go from what concentration to what concentration? High to low. Good. All right, the next bit, we're not going to spend a lot of time on here in lecture because we did talk about this in lab. You have to know the functions, right? So again, we talked about the inclusions already. Those are just those little chemicals that are part of what we call the cytoplasm, but they're not organelles. Cytosol is the fluid portion. Cytoskeleton is what supports the organelles. You can see this little protein network here. Centrioles we need for cell reproduction. They create the spindle. So again, cell reproduction for centrioles. So they make spindle fibers and cell division or cell reproduction. You could just highlight cell division there if you wanted. So cilia moves materials over the cell surface. Highlight that last one there. Flagella, we said, moves the cell itself for locomotion. Microvilli, remember we said, increases the cell surface area for absorption. And again, I always have these little notes underneath, so if you need more information, make sure you look at my notes. Sometimes those will give nice examples of this stuff on a PowerPoint. Ribosomes are where proteins are made. So sites of protein synthesis. We know that's where transfer RNA and mRNA meet, and we link up those amino acids. So protein synthesis happens at ribosomes. The endoplasmic reticulum, remember we have the rough ER and the smooth ER. The rough ER just modifies the proteins that are made on the surface by those ribosomes. So proteins are produced and modified by the rough ER. The smooth ER, its job is to manufacture lipids. You don't have to know about the cisternae. We'll talk more about that when we get to the metabolism and muscle unit. But smooth ER is lipids, rough ER is working with proteins. The Golgi apparatus works with both the smooth and rough ER, so they package those proteins and lipids made by the ER, and they decide if they're going to be used inside or outside the cell. So the Golgi is packaging those proteins and lipids into little vesicles to be used inside or outside the cell. So here we can see the rough ER is the proteins are made by the ribosomes on the surface. The rough ER modifies them, packages them. They're taken up by the Golgi apparatus here. You can see this one, their membranes fused. The Golgi apparatus packages it into a secretory vesicle, and then it's exported by what process would we call that? Leaving the cell through a vesicle? Exocytosis. Very good. Lysosomes is any vesicle that has a digestive enzyme in it. So they contain enzymes that digest other materials in or out of the cell. So they're kind of like the janitor of the cell. They have these digestive enzymes that can break down and clean up the cell. No, definitely not. Just digestive enzymes. So the pancreas, for example, secretes digestive enzymes to the blood. And they enter, I'm sorry, to the, no, not to the blood, to the pancreatic duct. And the pancreatic duct takes it to the common bile duct, which takes it to the duodenum, which is the first part of the small intestine, and that's where a lot of our digestion occurs, is in the first part of the small intestine with the help of those lysosomes from the pancreatic cells. That's why people with pancreatic cancer lose weight very rapidly, because they can't digest food because their pancreatic cells are cancerous and not functioning properly, and they can't absorb nutrients. Mitochondria, these are the energy producers of the cell because they make ATP. So when we talk about all the ATP needed for our sodium potassium pumps, if our mitochondria can't get the oxygen they need through our breathing and through our blood vessels working properly and our heart pumping correctly, then they're not going to be able to make ATP, and those pumps are going to stop, and then death occurs. So that's why we need oxygen, is to make ATP to cycle those pumps that maintain life. The nucleus contains the DNA and chromatin. Chromatin is DNA. So this whole structure here is the nucleus. So it contains DNA. It's not really a thing, it's a space. It's like your garage. Your garage doesn't do anything, but it contains your car, if you have a garage. And your car is what does all the work, right? So the nucleus is just a space, and what it contains is what's important, and that's the DNA, and then it also contains that little nucleolus that makes ribosomes. So the job of the nucleolus is to make ribosomes. So remember, the nucleus has this membrane around it with pores for things to come and go. What slips through the pores, though, that we already talked about? mRNA. Very good. Very good.